Now that we're recording, I'll do some announcements. It's nothing new, otherwise I wouldn't do it before class. And plus it's being recorded, so everybody will get to, well, anybody who watches the video will get these announcements. So to remind you, these are all reminders. We have lab today, unless you've heard otherwise from me, be here at noon. The lab we're doing is online, so make sure you bring a computer. I'll have some, but they're really crappy. So if you have access to a computer, you want to bring yours. I'm telling you, the ones we have, they're not very good. So yeah, make sure you're here at noon, unless you've heard otherwise from me. If you show up late, you get half credit. You know, invest for your lab. If you don't show up at all, you get 0. Or sorry, let me rephrase that. If you show up late, you get 3/4 credit at best. If you don't show up at all, you get half credit at best. So you know how it is. Make sure you're here. And it'll be like that for the rest of the semester. And that's another reminder. For the rest of the semester, even when we have online labs, we're going to physically show up unless you hear otherwise from me. So, you know, it always helps to take some initiative. So even though we're showing up in person on every Wednesday, if you see the lab and you look at it and like, wait a minute, I can do this before we even show up, go ahead and do it, because you might get handsomely rewarded for it, either by extra credit or maybe you won't have to show up, something like that. Any questions about today's lab? All right, and also it's pretty easy, I think. My opinion. I'm just hoping it was easy. What's the other reminders? The exam is posted, so you can retake exam 2. You have nothing to lose. If you get a lower score, we'll just keep your higher score. The retake is not timed. The retake is open book, right? So you can just like open it today and work on it all weekend, or for a week or 2 weeks. I don't care. You know, just make sure you email me when you're done, so I know to adjust your grade. Let's see. Do some attendance. See you wearing your WBSU gear. What's your credit there? Some other reminders, speaking of which, you know, if you're online and you can't do this WBSU extra credit, you can still take pictures. And I posted about that on the Facebook group. So, yes, sir.
When we go to other games, when we go to home games, we support, we can.
Yep, yeah. If you go to a game and you take a picture that proves that you were at the game, like instead of, I'll get you in a second. If you go to a game and, you know, the picture needs to somehow prove to me that you were there, not just like, I don't know how to put it, like it's you in the stands and like all I can see is like 3 people behind you. Somehow show me the game, so I'm like, oh yeah, that was that home game when they were playing so and so. So do that, and then if you have any other questions, email me because there's other ways you can make extra credit by doing that. Like if you share it to social media or if you're wearing your WBSU gear and all that stuff. So yeah, yes, ma'am.
What if I'm technically playing in the game? Do I get
I don't know. Let me think about it. I've been thinking about that, but now that you've asked, let me really think about it. I don't know. Let me think about that. Get back to you. Question online? Okay. Anyway, so that's it for reminder announcements. This is a reminder too. I just want to show it one more time. This on the screen. I keep telling you, please read my announcements. Some of you really, really could use some points. And I did a little search on my announcements, and it's been 17 times where I posted something and said, if you read this, email me back and tell me you read it, and you'll get some extra credit. 17 times I've done that. So, you know, you're leaving points on the board, and that tells me you're not reading my announcements. So some of you, too, have a really, really low grade, and some of you, luckily, are asking me what you can do. Well, first of all, read my announcements, not just with extra points, but I'm telling you what you can do. And these announcements are like, hey, don't forget to do this. Hey, don't forget this is due. Hey, here's some extra credit. Hey, here's some independent work ideas, right? Read the announcements. There's points there for you. Anyway, let's jump into it unless there's some questions. All right. Obviously, if you think of any questions, let me know. I'm gonna get rid of this. Those extra credits there for you, you know, not much, but you get an extra credit for doing what you should do, which is read the announcements that I'm posting. Let's get back to this. So remember, this first bullet point here is what we finished yesterday, and we just jumped into the second bullet point. When we were talking about this first bullet point, let's get this up 1 second, bear with me, please. There we go. All right. It took a long time to get through that first bullet point. And when we were talking about things in this first bullet point, I didn't give it a name, but what we were talking about, I'm going to abbreviate it here, was complete dominance, right? When we were talking about 2 different alleles, one being dominant and one being recessive, and all those examples, if you have the dominant allele, the other one was just completely masked, right? So those pea flowers, for example, if there was an allele for the purple and an allele for the white, then the flower would turn out purple, right? The white didn't even matter. Same thing with the way we said things work for, eye color in lab, right? We said if you had an allele for brown eyes and an allele for blue eyes, you would have brown eyes, right? Because that allele was completely dominant. Then yesterday, we finally jumped into what we call variations on Mendel's laws. So not everything is as simple as complete dominance. So the first thing we learned about was incomplete dominance here. And by the way, the first word for attendance will be plants, since it's up there, why not? Anyway, we learned about incomplete dominance. And all you really need to know for the exam, remember, you don't need to know all the examples. I'm going to ask you in the exam, I'm going to say this thing made it with this thing. These were the offspring. What kind of dominance is that, right? So if I said a red flower and a white flower mate, and all the flowers, all the offspring are red, that would be complete dominance, right? Because red was dominant to white. Then we talked about what we had yesterday. So in that case, I would say if a red flower mates with a white flower and the offspring are pink, then that would be incomplete dominance because that's a mixture of the two, right? It's a mix of those two phenotypes, those two alleles. Next thing we're going to talk about is ABO blood groups, and that's the way your book presents it. But let me just go ahead and tell you this. I don't know why. This should be titled codominance, because ABO blood group is just an example of codominance. And what you need to know about is codominance. I'm not going to ask you any questions about ABO blood groups. I don't know why they did it that way. They should have called this section codominance and gave you some examples of it, and one of which could have been ABO blood groups. But let's talk about it. Oops, we already done talking about incomplete dominance. I'm a little bit behind. There we go. ABO blood groups. So you don't have to take notes on this, but I'm going to go ahead and kind of briefly explain it since your book did. ABO blood groups involve 3 alleles. So this is also more interesting and different than everything we've looked at. So far, everything we've looked at has involved 2 alleles. It's either this or that, right? Purple flower or white flower, red flower or white flower, blue eyes or brown eyes, widow's peak or no widow's peak. It's always been 2 alleles. Here, it gets a little bit different. Here we have the 3 alleles, and the 3 alleles give us 4 different phenotypes, and they happen to be blood types. I'm sure you've heard of them. You're either A, B, AB, or O. So even though it's not on the slide, basically when we're saying there's 3 alleles, the 3 alleles we're talking about are A, B, and O, right? So you're going to see how this works as we move forward. But basically, these letters refer to carbohydrates. So if you're ever wondering what your blood type means, now you're about to find out, right? So if you're you're blood type A, that means you have the A carbohydrate on the outside of your cells. And I'll explain this in a second with a picture. If you're B, you have B carbohydrates on the outside of your blood cells. If you're AB, you have AB carbohydrates on the outside of your blood cells. And if you're O, you have none of them. That's what I was saying, right? If you're A, you have A. If you're B, you have B. If you're AB, you have both, and if you're O, you have none. So I'm going to put a big X to this too when I'm discussing it. I'm only going to explain it because I don't know. I guess it's kind of good to know, but mostly because the book includes it. So here we go. The different blood phenotypes. Again, A, B, AB, or O. The thing is, A and B are both dominant. They are codominant, and I'm going to explain more about codominance as we move forward. But unlike complete dominance, right? If A was completely dominant to B, then what would happen is if you had A and B, like this one does, AB, sorry, I know you guys can't see online. If you had the AB and A was completely dominant, which it's not, then everything would be, you'd only have the A carbohydrate, but it's not the case, right? This is codominant. So in this case, if you have the alleles from both of your parents, so 2 different alleles, then you have both phenotypes. That's the most important thing as far as what you need to know is concerned. But despite that, I'll kind of explain how it works physiologically. So if you're blood type A, again, that means you have the A carbohydrate on the outside of your cells. That means your body is not like the B's, right? If it gets any B blood cells, it's like, whoa, those shouldn't be here. That's a foreign, that's a foreign blood cell, and it attacks it. So if you are blood type A, that means you are anti-B. So if you're getting blood, you cannot get B blood, and you can't get AB blood, because even though AB has your carbohydrates, it also has the B carbohydrates, and you are anti-B. I'll explain it later. So blood type B, it's kind of like blood type A, except it's anti-A. So if you're blood type B, you can get O, which I'll explain later. You cannot get A, obviously, because you're anti-A. You cannot get AB, again, because you're anti-A, but you can get B, obviously, because you're B. Now, AB is slightly interesting because it has both. And because of that, it's not anti-anything. This is what they call a universal acceptor. So have you ever heard the term universal acceptor? You don't know. Well, hope you guys are looking this stuff up. I know everyone's got the facing screen, but hopefully it's about biology. Anyway, AB means you're both, right? So because you got both carbohydrates, you are the universal acceptor. You can take A blood because you're not anti-A. You can take B blood because you're not anti-B. You can take AB blood because you're not, you are AB. And then finally we have O. That's what I happen to be. That's the universal donor. So with O blood type, that means you are anti-everything. So I've got some hateful blood, right? I like to take as O. I don't like A's, I don't like B's, I certainly don't like AB's. So that's how that works. Any questions about that? All right, moving forward, you don't have to read this. I'm not going to ask you any questions about it on the exam. There might be questions about it on the study guide, but that kind of describes exactly why you are anti-A and anti-B. Yes, ma'am?
Okay, so O's can't take anything but O's, but I swear, like, everyone else can take O. Oh my God.
Exactly. Because O doesn't have any of the carbohydrates, it doesn't have A's, so it's good with B. It doesn't have B's, so it's good with A. So universal donor is O, universal acceptor is AB. Yeah. And of course, that's just A and B. It doesn't even tell you about the positives, right? AB positive, O negative, all that. So if you want to look up what the positive and the negative are for independent work, feel free. Like I said, this slide kind of describes exactly why you are anti-A or anti-B or whatever. You can read it if you want. The four blood groups results from various combinations of 3 different alleles, like I already said. So this is everything I've already said, but it's in writing. Again, if you have the A blood group, you make the carbohydrate A, or A allele. If you have the B allele, you make carbohydrate B. If you have this little I, which again, remember, that's recessive, that means you don't have A or B. Now, moving forward. Here's what we really need to get into, which is this word codominant. Like I've already said, what we're really talking about, the important part for what you guys need to know for the exam, we were talking about codominance. I'm still going to skip this slide and get to what you really need to know. Here's some good examples. Some non-book examples of codominance. This is stuff I came up with. So if you had a black dog and a white dog, then you would end up with a black dog with white stripes, or maybe a black dog with white spots, or a white dog with black spots. Now think about this. I want to make sure you understand this. If we were talking about complete dominance, if black was, dominant to white, then if a black dog and a white dog made it, you would have a black dog. Does that make sense? If it was complete dominance, the first thing we talked about. If it was incomplete dominance, which is the last thing we talked about before we ended on Monday, and black was dominant to white, actually it wouldn't matter. If you mix a black and a white dog, you would get gray, right? Because it's blending the two. Now, codominance was both phenotypes showing up. So black with white spots or white with black spots. Or a red cow with a white cow. And that actually does happen. There's a name for it. Instead of it being reddish white, it would be white with red spots. Or, white with red spots. Because it's both phenotypes showing up. It's not a blending of the two. It's both. Hence codominance. Again, a red flower and a white flower, that means you would have a red flower with white spots, or even a white flower with white spots. A white flower with red spots. The idea here, though, again, it's both phenotypes showing up. So in the exam, someone's going to miss this, and it shouldn't be that hard, especially since I'm saying it over and over again. I'm going to say this thing mated with this thing. I don't know what yet. It's going to be hypothetical. It might be completely made up. I don't know yet. This thing mated with this thing, and these were the offspring. So if I say this thing mated with this thing, and the offspring had both phenotypes, not a mixture of the two, but both phenotypes, that means it is codominance. So just like if you were, co-parenting, like if you're divorced and you're co-parenting, that means a lot of times, usually what that means is the kid lives with both parents separately, right? Both parents. It's not blended like if they were living in the same house. It's separate with both parents, right? So just like this, both phenotypes are showing up. It's not a blend of the two, it's both. So any questions about what codominance is? All right, so we talked about complete dominance, we talked about incomplete dominance, and we talked about codominance. In my opinion, those are the easiest. However, those are not only the easy, but I guess one might argue that they're also easy to kind of mix up. But anyway, let's move on to something a little bit more complicated, and that's called plyotropy. And if you guys have watched that video that I posted in the last lab, the genetics lab, then everything I'm about to tell you is a review. This guy, Hank, talks about it, and I highly recommend watching it if you haven't yet. Plyotropy. That is when one gene influences several characters. Make sure you know that. And again, in the exam, I'm going to make up some crazy off-the-wall, not even a real situation. But if I describe something where I'm talking about one gene and then I talk about multiple characters, that's plyotropy. And maybe some of the ways that'll help you remember that is the P or the PL. Maybe you could think PL. Also, plural starts with PL, right? Plural, like more than one. So hopefully that'll help you remember. Plyotropy means there's one gene with several characters. Make sure you know that. The next word for attendance, I'm going to circle it. I'm not going to say it. So if you're online, make sure you look at the screen. I've circled the next word for attendance, and I'm pointing an arrow at it. Anyway, speaking of plyotropy, your book gives an example, and it is sickle cell disease. I'll go ahead and put it next to that, not because you don't need to know it, or because I'm probably not going to ask you questions about sickle cell disease on the exam, at least not in the context of heredity. We will talk about sickle cell disease when we talk about evolution, when we talk about mutations and things like that, but for now, this is your first introduction. Anyway, sickle cell disease, like I said, it's an example of plyotropy because it's one gene, and that one gene causes abnormal hemoglobin proteins, right? So remember, to remind you what a gene is instructions on how to build a protein, right? So there's one gene that's messed up, right? There's an allele that's messed up, so the hemoglobin proteins are messed up. And because of that one thing, that causes the red blood cells to deform into a sickle cell shape with jagged edges, and then that leads to a slew of other characters, if you will, or problems, which would be here, right? The sickle cells can lead to a cascade of symptoms such as weakness, pain, organ damage, and paralysis. So that one gene, that one allele, right, the one allele of that gene, causes all these different phenotypes, if you will. Symptoms, phenotypes, in this case, those two words mean the same thing. Because this is the physical manifestation of the genotype, right? If you have that one allele, then you have these symptoms. And again, that's just an example. I'm not going to ask you any questions about sickle cell. And that's it for plyotropy. Are there any questions about plyotropy? So remember, plyotropy kind of sounds like plural. So there's one gene and many characters. That in itself isn't that difficult, right? Well, let's make it a little bit more complicated. The next one we're going to talk about is polygenic, I should say polygenic, not polygenetic, that's a typo. Polygenic inheritance, which really sucks because I just told you the PL should remind you of plural, and now I'm giving you another P word. And not only that, but the P word starts with poly, which means many, right? So I'm about to make it more complicated. Polygenic inheritance. It's kind of like the opposite of plyotropy. In this case, it's the one character, but more than one allele, more than one gene, more than one gene. So instead of having like sickle cell, you have that one gene that can cause all these different characters, all these different symptoms. Here, we're talking about just one character and a lot of genes that are causing it or affecting it. Does that make sense to everybody? All right. For the exam, this is an important bullet point. There's a question on the study guide that will really make sense to you. Basically, it says something like, if you're trying to determine if a character is caused by polygenic inheritance, what do you look for? And one of the answers is, or one of the options, the correct answer is the bell curve. So meaning when you graph it, if you were to graph a bar graph, it would be a bell curve. Unlike everything else we talked about. So, like if we took a graph, we did this in lab, we just didn't make it into a graph. If we made a graph of the number of people, like eye color, we said in this class, after we did the lab, there was, I don't know, however many that is, that many people with blue eyes and that many people with brown eyes, right? That's a graph. Is that a bell curve? No. First of all, you need at least 3 to have a bell curve. I mean, more than that, really. That's just a bar graph. But a bell curve would have a lot of bars, and it would look like this. Because you're looking at more than just 2 characters, or excuse me, more than just 2 genes, and the character has a lot more variation. So your book is using height as an example. And of course, height is actually a lot more complicated than this, and this is a simplified version of height. So far, everything we've talked about have only been 2 genes, right? So sometimes we talked about, I don't even want to say that. This is the first time we're talking about something with 3 genes, right? So we're talking about 3 genes affecting 1 phenotype. So because of that, we can see, we have all these different phenotypes available. You have very short, medium, very tall, everywhere in between. Of course, I'm going to put it next to this because I'll remind you this is just an example. When you're studying, don't feel like you need to memorize anything from this slide. Yeah. So yes, polygenic inheritance, multiple genes affecting 1 character. That's what you need to know. And height does have multiple genes affecting 1 character, and it's more than just 1. Also, there's a lot of influence from the environment. Anyway, speaking of which, that would be the next slide we talk about. Any questions about polygenic inheritance? All right, the next thing we're talking about is epigenetics. And again, like I said, with height, you could have, for example, 2 identical twins. So obviously they have the same exact genes, the same exact alleles, right? So you would expect to have the same height. And usually they do have the same height. However, especially if you were to separate those genes, separate those twins and have them live completely different lives, then they would be slightly different heights because of epigenetics. And let's talk about what epigenetics is, which you do need to know. Many characters result from a combination of heredity and environment. So basically, epigenetics is when we're also not just looking at the gene, but we're also including the environment. So when you think of epigenetics, you should think about environmental influence on genes. So whether or not these things are more influenced by genes or environment, or as you probably heard it called before, nature versus nature, it's a very old and probably contested issue. You can look, there's a lot of stuff you can look up for that for independent work if you're interested in controversial stuff. Like people might say, is homosexuality, is that a gene or is that a choice? Or excuse me, was it in your genes or was it the way you were raised? Or any other thing. Just think about anything you want to think about and just look it up if you want. The musical abilities, is that in your genes or is it the way you were raised? Being a serial killer, is it in your genes or is it the way you were raised? And again, like I said earlier, when we were talking about height, I gave the example of identical twins. How many of you have ever I've met identical twins, but I don't have any friends. I've not spent a lot of time. You have? So do you notice it? Do you notice some physical differences between them?
Yeah.
Yeah, even though they're identical, right? It's because, it's the environmental influence. What I haven't told you yet, this is going to be very important in the next chapter, so this is a preview. I keep saying that genes are instructions on how to build proteins. What I haven't told you yet is that your chromosomes are almost like books, right? They're books of instructions on how to build a particular gene. But it takes a little bit of something to get to those genes, right? So sometimes the genes are wide open and ready to read, right? So you're building a lot of those proteins. Sometimes it's harder to get to them, so you're not building a lot of those proteins. Sometimes the environment tells your genes, all right, I need this protein, right? So if you have 2 identical twins in 2 separate environments, one might be in an environment that says, all right, we need to make this, and it kicks on that gene. And the other one might not be in a different environment, and it's not turning on that gene. So even though they have the same set of instructions, it doesn't matter unless you turn them on. Remember we were talking about the cell cycle? We had the on-off switch. It's kind of the same here. Just because you have genes doesn't mean they're always working. So the environment is turning genes on and off. So again, even though you might have identical genes, like identical twins, that say build this and build it the exact same way, if one's being told to build it and the other one's not, they're going to be slightly different. Your book also points out that in general, it's only the genetic influences that are inherited, right? So everything we've talked about so far, when we're talking about inheritance, has been genetic, right? It's what allele you have. The alleles that you got from your mom, the alleles that you got from your dad, that's what's inherited. And generally speaking, it's not the effects of the environment that are passed along. So, all right, you have to really rack your brain and try to remember this one. I told you this would come back. When we first talked about, I think, chromosomes, I said the DNA wraps around these little balls called histones. And that makes something called nucleosomes. And I told you that was important. And the reason why, and I'm really oversimplifying things, but hopefully this is close enough. When the DNA is wrapped around those balls tightly, basically, then the machines that need to get on the DNA and read the code, it's just harder for them to get to that, right? Because they're wrapped around the ball and it's tight. But sometimes you do things, the environment makes it different. And it makes it, I don't know, open and easier to get to. So then the machine that reads the genes can get to it and read it easier. And that is what epigenetic inheritance is. Well, one of the things too. Basically what I'm saying is it's not just how the environment influences your genes, as in tells your cell you need to make this protein, you need to make that protein, right? It's not just that, but sometimes it can make changes to your histones, basically, making them harder or easier to read. And sometimes we found out that that can be inherited. This is a lot of information. So I'm going to put it next to this. You can read it. You should read it. Matter of fact, even for those of you here and not online, you also have a word, an attendance word. Not that you have to, but for extra credit, when you get a chance today, send me that word. Whoops. Leave back out of here. No, that'll work. Anyway, so all this to say, you should read it. But really, when it comes down to what you really need to know for the exam overall, when we talk about epigenetics, again, it's the environmental influences on your genes. So, again, the environment tells your gene, your cells, when to make certain proteins. And sometimes your environment changes your chromosomes, not your genes, not the letters, not the A's, the T's, the C's, and G's, but the way your chromosomes are wrapped up and put together. Sometimes the environment changes that. Sometimes that change can be passed down to the next generation. So that's it for epigenetics. Oh, come on. Doing something. There we go. And basically that's what this next slide says, right? Epigenetic modifications, which again is the change to basically how your chromosomes are wrapped up, and changes in gene activity that results may be carried on to the next generation, which is a little bit of a scary thing, because that means you could screw things up, right? You could mess things up by doing things in your life that might be carried on to the next generation. And unlike alterations to the DNA sequence, which again is the A's, the T's, the C's, and the G's, the actual code, unlike that, the chemical changes to chromosomes, depending on what they are, as far as we know, can be reversed. So that's the good news. Can fix some of the things you've done, so to speak. So any questions about epigenetics? Awesome. That means we are going on to the third and final of the main bullet points, which is the chromosomal basis of inheritance. And the good news is I can go through a portion of this kind of quickly, because as I've been talking about everything else, I have been talking about this in the context of chromosomes. Excuse me, your book hasn't, but I have. So what I'm about to tell you is basically a review of what I've said, just with some pictures. Except for the sex determination and sex-linked genes, that'll be slightly new information, but let's talk about the chromosomal basis of inheritance. The chromosomal theory of inheritance states that genes are located at specific positions on chromosomes. I've already said that. That's nothing new, right? So even though that's on the board, there's no need to write it down. So we learned that on a previous lecture, right? Genes are located at specific positions on chromosomes. So if, again, for example, I wish I could give you a real example. I'm trying to remember the number from that video from Hank that I shared with you guys. There's a gene that determines whether your earwax is wet or dry. I think he said it was on chromosome number 6 or something. So for all humans, you could look at chromosome number 6, if I'm correct, at the same spot on the same chromosome for every single human, and that is where you will find that gene, right? That's what that's saying. And the behavior of the chromosomes during meiosis, when those alleles split, and fertilization, when they come back together, that accounts for inheritance patterns. There's no need to write that down either, because we've already learned that. This is all basically just reminding you of everything we've said, right? You have genes on specific chromosomes. Those homologous chromosomes split during meiosis. They come back together in fertilization. The way that thing happens determines what you get, right? What you get, what you inherit. So it's the chromosomes, and again, you should already know this, it's just a review. It's the chromosomes that undergo segregation and independent assortment. So your book keeps saying these alleles go through independent assortment, and these alleles go through segregation. But really, because these alleles are on the chromosomes, it's the chromosomes that do it. And it's the chromosomes that count for Newton's law. But again, I've been saying this the whole time. So even though your book hasn't been focusing on it, I have, so this really should be a review. Which is this as an example. So we talked about this dihybrid cross earlier, where we were looking at a purebred red, round, yellow pea plant versus a purebred wrinkle green pea plant, and then you cross them, right? You get your dihybrid cross, and then you have your offspring, and then they're all round and yellow because round and yellow is dominant. Blah 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 blah. The point here is, last time you saw this, your book didn't have little pictures of chromosomes. So it's the same thing. So now instead of saying, these chromosomes separate in meiosis, we're just these alleles segregate in meiosis. We're saying specifically it's the chromosomes that the alleles are on. But nothing's new here. This is the same information. We're still talking about alleles segregating, right? So that's what's happened here, right? This has a big R and a little r. This has a big Y and a little y. That's one of its somatic cells, but then it goes through meiosis, right? And it splits them. So the big R and the little r split, right? And the big Y and little y split. Well, we haven't got to the split yet. They line up independently, right? So what we're saying here is there's two ways these could line up. We could have all mom on one side, all dad on the other, or maybe we'll have dad and mom or mom and dad, right? That's independent assortment. So this time it's the same thing I've already told you, except now we're not just talking about the alleles, we're talking about the chromosomes themselves. So when this plant makes a sperm or an egg, these are the two possibilities for those chromosomes, right? That's the two possible ways they can line up. And then of course, once they segregate, that means we have these 4 different possibilities after it goes to meiosis for that, and these 4 possibilities after meiosis for that. We'll put a big X through this because when you're studying, there's nothing in the details that you need to know for this, right? This is just a big, complicated way of reminding you that these things first go through independent assortment, right? Meaning they line up independently of each other. There's no rhyme or reason for the way they line up. And then they segregate. That's all it's saying. And it's not just the alleles that do this. It's the chromosomes that those alleles are on. What this should show you, if I had the small enough writing to do this, is if there was another gene on here. Let's call it Q. What if there was a Q right there? Q, the gene Q on that same chromosome, whatever chromosome that is, if there was a gene Q. So even though R and Y are independent of each other, they are, right? Because they are on completely separate chromosomes. Here, this one, when it makes eggs or sperm, whatever, here, in this case, for both of these chromosomes, one gets the mom chromosome, one gets the dad chromosome, right? So another way of saying that is one gets big R, big Y, the other one gets little r, little y. Here, if you were to do the same thing, let's see, how about a little q right there, big Q right there. In this case, when these things segregate, you would get little r, little q, even though it's not drawn there, big R, big Q. The point here is that what I'm showing you is independent assortment, but if they're on the same chromosome, it's not independent, right? So here, when these things segregate, no matter what, if you're getting a little r, you're also getting a little q. I should say no matter what. As far as we're concerned, if you get the little r, you're getting the little q because they travel on the same chromosome. And here, this one got the big R, therefore it also got the big Q because they're all on the same chromosome. Whew. Anyway, and then there's all the whole pictures all together. So let's move forward and formally talk about what I just talked about, which is linked genes. So what I just described to you was something called linked genes. Here's a good example from your book, not from your book. So we don't know what these genes are, gene 1, gene 2, gene 3, gene 4, right? They're all on the same gene. So when you go through meiosis and you're making sperm or egg, whichever one that is, let's say that's your mom chromosome. If that's the mom chromosome, then you're going to get the alleles for all gene 1, gene 2, gene 3, gene 4, those are all going to be mom alleles because they're all on that mom chromosome. That makes sense? They all go together. So if your kid gets your mom chromosome for, I don't know, number 7, I'm just making that number up, then it's going to get all those alleles. Does that make sense? So instead of saying like, I don't know, I got my mom's, I don't know, my mom's allele for eye color and my dad's allele for toe shape, right? I'm just making that up. Those are 2 different alleles, and I got one for my mom, one for my dad. How does that happen? Well, one's on one chromosome, one's on the other chromosome. So when they segregated, you know, like I got one, happened to get a mom chromosome for that one, happened to get a dad chromosome for that one. Remember, independent assortment. There's no rhyme or reason to which one you get. But whatever genes are on this mom chromosome, they're all going to travel together. So if that one, gene 1, happens to deal with, I don't know, if you have gene 1, you know, in a certain allele, you could have red hair, and gene 2 means freckles, which is not necessarily the case. So if you got the mom chromosome that says you have gene, you know, freckles or red hair, then you'll also have freckles. Point is, they travel together when they're linked. To make things even easier, and I hate to do this, but for the exam, it's going to be really easy. When they ask you what linked genes are located near each other on the same chromosome. That's really just that simple as far as the exam is concerned. What makes it a little bit more complicated, again, because they travel together, then you're not going to have the 9 to 3 to 3 to 1 ratio that I told you about earlier. And also, think about this. I'm going to make it even more complicated, even though I'm not going to get this complicated on the exam. That's the mom chromosome. What if the dad chromosome is right there? And then it'd be crossing over. So then you might have gene 1 from mom, gene 2 and 3 from mom, right? But then it swaps, it does crossing over. So that chromosome, you have these 3 mom genes, and then 1 from dad. Meanwhile, not shown here would be the dad chromosome that has gene 1, 2, and 3 from the dad, and then gene 4 from the mom. So that makes it even more complicated. And your book gets into it really specifically. It gives you a bunch of weird numbers. So if you get anything other than 9 to 3 to 3 to 1, basically, the Cliff Notes is that means it was linked genes. I think there is a question like that on the study guide. So that's what you do need to know for the study guide. If you get anything, it'll still be 4 digits. You still get a 4-digit ratio, but if it's not 9, 3, 3, 1, that means they're linked and they did not travel independently because they were on the same gene. It's crossing over occurred. The next word for attendance will be, let's see, located. Located is the next word for attendance. Any questions? All right, here's probably the easiest part of this chapter. Sex determination in humans. This is pretty straightforward. Many animals, including mammals, have a pair of sex chromosomes that we already learned about in the last chapter. They're designated as X and Y that we already learned about in the last chapter. Males have an X chromosome and a Y chromosome, just like we learned in the last chapter. Females have 2 X chromosomes, just like we learned in the last chapter. So we've talked about it for 2 different chapters now, and yet someone's still going to miss this question. Again, I'm going to be sad about it, because it's pretty simple. Males have an X and a Y. Females have 2 X's. That's basically it, right? So I guarantee you there'll be questions about that on the exam because I've given it to you for 2 different chapters. Males have an X and a Y. Females have an X and an X. And it's like that for all mammals. What is really interesting is when you look at other species. If you look at, I don't know, fish or insects, it gets a little bit more interesting. It's not as straightforward as mammals can be. Anyway, any questions about sex determination in humans? There's a picture of an X chromosome and a Y chromosome. There you go. The last thing we'll talk about in this chapter is sex-linked genes. What is a sex-linked gene? Well, it's something you should know for the exam. It is a gene located on a sex chromosome. It's kind of in the name, right? Which is interesting when you think about it, because everything else we were talking about has 2 alleles, right? So, because they're all on the same chromosome. But here, if you're a male, you don't have 2 of the same chromosomes. You have an X or a Y. Whereas if you're a female, you have 2 of the same chromosomes. But for a male, you don't. And that makes things a little bit more interesting when it comes to sex-linked genes. And we'll talk about why here in a second. But there any questions so far, at least what a sex-linked gene is? Not necessarily how it works, but what it is. All right. Let's talk about how it works. Your book gives some real life examples, including red, green color blindness, hemophilia, a certain type of muscular dystrophy. All these things result from a sex-linked recessive allele. I'll put it next to this, because that's nothing you need to know for the exam. That's just the authors of your book saying, hey, this is important because these are real life things. It's not just like some academic discussion. These are real things that really affect people. Red, green color blindness, for example. You can read all about that if you want. I'm not going to test you on it, but it is very common. Obviously, it's a sex-linked disorder. That's why we're talking about it. And specifically, it's caused by a malfunction of light-sensitive cells in the eyes, right? So there's something wrong with your eyes, the cells in your eyes. And the interesting thing about this is it's mostly the males that are affected. Not that I'm going to ask you that, but it's mostly males, and we'll talk about why in a second. And hopefully, if you're a thinker, you're already starting to think about possibly why. I just said earlier, males have an X chromosome and a Y chromosome. So for some things, or for everything on sex-linked genes, they only have one version, right? You either have a Y version of something. There's genes that are on the Y chromosome, and there's genes that are on the X chromosome, and they're not the same. Anyway, can anybody not tell me what number that is? You can't see it?
No.
Interesting.
7.
Yep. Anybody else?
Yep, 7.
Yep. Can you see it? Interesting. Can you see it? He's watching video. Not even close to
Come
it.
on, for
Anyway, can you read that number? No? Interesting. I can read it. So you guys have you had your eyes tested? Do you know if you're colorblind or not?
I had vision of you.
Okay. How about you guys? You might be colorblind if you can't see that. That's a 7.
So wide, right?
Yeah, it might be too bright. Yeah. You might not. Yeah, I mean, it's easier to see right here.
Oh, yeah. Yeah, that's way.
Oh, okay.
That's different.
All
That's
right.
different.
No, nothing to worry about then. You don't need to write home and say I'm colorblind.
That's colorblind.
But because they're located on the sex chromosomes, sex-linked genes exhibit unusual inheritance patterns. That's basically what we're going to talk about. And here's where it gets slightly interesting. All right, so this is on the X chromosome, right? Remember, males only have one X chromosome. So here we have a mating between a female with completely normal vision and a male who's colorblind, because this is a recessive disorder, right? So the female goes through meiosis, she produces some eggs, whatever, 100% of them are going to be the dominant, meaning you can see just fine. The male, when he goes through meiosis, half of the sperm are going to be Y chromosome, the other half are going to be that one little N, right? The one that actually gives you the disorder. So those are the possible outcomes. If you have a colorblind male with a normal non-carrier female, 100% of their kids are going to be not have colorblindness, right? But here's really quick before I move forward. This is why this works out. And this makes it a little bit more complicated. Remember earlier I said, for all this chapter I said, to have a recessive phenotype, you have to have 2 recessive alleles, right? What I really should have said, but I didn't like to confuse you back then, the more accurate thing to say is if you have the recessive phenotype, then 100% of your alleles need to be recessive. Which is almost saying the same thing, because if I said, you have 2 recessive alleles, that's the same thing as saying 100% of your alleles are recessive. But in this case, he only has 1 recessive allele, but that's the only one there is, right? There is no other N allele. So 100% of that dude's alleles are recessive. It's not 2 of them, but it's still 100%. Does that make sense? There is no N on the Y. There's only an N on the X chromosome. So because of that, he's colorblind. He has a recessive disorder, even though he only has 1 allele, because he still has 100% of the alleles. Anyway, here's a different outcome, or a different version. Here we have a normal male mating with a carrier female. So she has the allele for colorblindness, which is not 100% of her alleles, right? It's only 50% of her alleles. Therefore, because it's a recessive disorder, she doesn't have it. She carries it. She goes through meiosis. 50% of her sperm have the normal allele, egg. 50% of her sperm have the recessive allele. Obviously, when this person goes through meiosis, 50% of his sperm are the dominant allele, and 50% of the sperm are just Y, and don't have that allele at all. So then there's a possible outcome of that. There's a 25% chance that they'll have a boy with color blindness, a 25% chance that they'll have a female carrier, a 25% chance that they'll have a male, normal male, and a 25% chance that they'll have a female, normal female that's not a carrier. I'm not going to test you on any of this. This is just all the different ways of looking at it. And your book is pretty thorough because here's yet another one. Did I hit the right one? Wow, that's hard to read, isn't it? It is. All right, what you can't see, this would be a lot better if you could, is this is actually shaded. It's not as dark as this, but it's shaded. Because now what we have is a carrier female mating with a colorblind male. What are the possible outcomes? Well, again, you go through meiosis, 50-50 egg, 50-50 sperm. You have a 50% chance of having a colorblind male. 50% chance of having a colorblind child. 25% specifically is a male. 25% is a male. 25% chance of having a completely normal male, and a 25% chance of having a carrier female, and a 0% chance, because it's not on here, of having a normal female. Because remember, normal female would be X, big N, X, big N. Now, I know I'm going through this kind of quickly because there's not going to be any questions on it. If you do need more help with it, let me know and I can talk to you separately. Or watch that video I posted in the last week's lab with Hank when he talks about genetics. He talks about this stuff. The next thing we're going to talk about, are there any questions about color blindness?
Yeah.
Yes.
I'm guessing it doesn't matter. Is it just the little n that means it's colorblind?
Right. It's a recessive disorder, meaning if you have the big N, then you're complete, you're fine. The big N is dominant to little n. But because the male again only carries the one allele, then that means if the male gets the little n, he has it because 100% of his alleles are recessive.
Right. So can the female get the colorblindness?
Yes, right. That's this case right here. So if these two people mated, this female carrier and this male colorblind person, there's a 25% chance that they would have a female who is colorblind. Because she would have to have both of those alleles to be colorblind, and that one is.
I'm guessing it's harder for females to be colorblind than a male?
Yeah, for that reason, right? Because this one, she has the one allele, but she's not colorblind because she has a functioning allele. Male only needs one of the recessive alleles. And the reason that works on a molecular level, basically, is because, right, let's just say this gene, whatever N is a gene that says make this protein that's going to be in the cells of your eyes, right? So let's just say the little n is broken. It says, it just doesn't work, so that means you're not producing those proteins. So as long as you have one of the big N's, then even though the other allele is broken, that allele, the big N, works, and you properly make the proteins, and then you can see color. You're not colorblind because you're making those proteins. Unlike somebody who does not have the big N, they're not making the proteins, therefore they can't see. Any other questions? Okay, the next thing we're going to talk about, I'm going to put a big X through this because there'll be no questions about it on this exam. There might be on the study guide, but hemophilia, I like that you broke those in here. Not that it's important for understanding biology, but it's good because hemophilia is actually important outside of biology. Your book points out that it's a sex-linked gene, sex-linked recessive trait, just like colorblindness, with a long, well-documented history. The reason it's well documented is because the age-old practice of strengthening international alliances in Europe by marriage spread hemophilia through royal families. Another way of saying there was a lot of inbreeding going on in Europe, right? That's how they strengthened the alliance. This is interesting for a few reasons. First of all, if you know anything about World War I, it wasn't necessarily just the assassination of Archduke Ferdinand. That was kind of like the match that lit the fire. Like people were building the fire before that because they were kind of moving away from this inbreeding. So that made things a little bit more unstable. But anyway, your book example is this, and I don't remember the exact details. You can look this up for independent work. We're not even specifically this, but you can look up at what time has biology influenced history, and this is one of those times. So basically, we have Alexis here from the royal family of Russia. Alexis had hemophilia because of that, they had a really weird guy on the grounds, Rasputin or whatever, because he was there to help Alexis. But he was a really weird dude, and all the Russian people thought that really weird dude is influencing our royal family, and because of that, we have the Russian Revolution. All because of inbreeding and hemophilia, right? Well, not all because of that, but that was a big portion of it. So look that up if you want. And again, like I said, any other time that biology has influenced history. That's a lot, especially when you think about battles. Like how many times have battles been lost simply because the other side had a disease, or because their leader was completely nuts because they had something wrong with their brain biologically. Anyway, any questions about that? And again, I'll put it next to that because I'm not going to ask you that. I'm not going to be a test question that said, was Alex or Lewis a carrier? And you'd be like, oh, well, obviously Alex was a carrier and Lewis was completely fine. I'm not going to ask you any of that. Anyway, if there's no questions about that, I'll remind you at the end of every chapter we do the evolution connection. I always skip it. It's kind of interesting. It's about dogs, how wolves turn into dogs, or how we turn wolves into dogs, so on and so forth. You can read that if you want. The last word for attendance is going to be right here. Nikita. All right. Nice. All right. And that's it for the recording. If anybody of you wants extra credit for the WBSU picture, you can stick by. We'll take a picture of the back corner. And if either body does not WBSU, want to take it? All right.